Hello and welcome to today's lecture on immunity to bacteria. I just want to remind you before we get started that all of our majority of our figures will be coming from your Janeway textbook, and you'll see the figure numbers at the bottom if you want to reference them for yourself back in the textbook. So pathogens that infect multicellular hosts come in a great variety of sizes and have very different lifestyles. And these sizes and lifestyles and types of pathogens will greatly affect the immune responses that hosts launch against these pathogens. Immune responses against pathogens vary depending on the time of infection and the burden of pathogen. So what happens at the establishment of infection is we have predominantly innate immune responses, and later in infection we have our adaptive immune responses. And today we're going to be focusing on these concepts of innate and adaptive immune responses to bacteria specifically. So we have both intracellular bacteria here and extracellular bacteria here. Now this lecture will build on your knowledge of different types of immune responses to show how different responses work in consortium to eliminate pathogens based on their PAMPs and their infection patterns. Today we'll be going through a very brief introduction to bacteriology, and we'll learn about what bacteria are and different types of bacteria that exist. We'll go through innate and adaptive immune responses to bacteria, and then we'll also talk about how bacteria have evolved to evade and subvert immune responses to establish infections. So let's start at the start and go through some very basic bacteriology. So what are bacteria in general? Bacteria are a very, very large class of pathogens that are unicellular organisms that have cell walls, but they don't have any organelles inside of them, and they do not have an organized nucleus. Now, there's a very large amount of different ways to classify bacteria. This can be by shape, by infection style, by size, and a variety of different other factors. But today we're going to be going through how different bacteria are classified in a way that is most relevant to how the immune response will either recognize them or fight them off. So first we're going to start with talking about the difference between gram-positive and gram-negative bacteria. So gram-positive and negative are called for this Gram test, which is basically a staining that involves crystal violet, iodine, and alcohol and safranin, such that gram-positive bacteria seen here are labeled purple at the very end. So these circular guys are purple and they're gram-positive. And then the gram-negative bacteria come up as red or clear depending on the stage of this test. And this is how the different bacteria are classified as gram-positive or gram-negative. Now, obviously this is just on one test, but there is some biology behind these differences. And the difference as to why some bacteria are stained positive or negative on the Gram test is based on what is contained in their cell walls. So if you look at gram-positive bacteria, this is just a schematic of the cell wall of the bacteria. Here you have our cell membrane with proteins and phospholipids and such. And gram-positive bacteria's cell wall specifically contains teotic acid seen here, and lipoteotic acid seen here. And this together, they also have lots of peptidoglycan. And this together is their cell wall. Now, gram-negative bacteria are slightly different, as you can see, and here's their cell membrane. And although they do have peptidoglycan seen here, they have this lipoprotein, and they have this large outer membrane that the gram-positive bacteria do not have. And this is covered with what is called lipopolysaccharide, or LPS. Now, this is a protein or lipopolysaccharide. You're going to want to remember that term, but this is the general difference between the outsides of the bacteria that are gram-positive and gram-negative. And keep in mind that you can start thinking about, okay, why is it important to classify the different bacteria based on their outsides of their cell walls? And maybe keep in mind that a lot of adaptive immune responses actually are targeted towards the outside of bacteria. Now, another way of classifying bacteria is the location of their replication, and we can differentiate bacteria based on intracellular or extracellular bacteria. So we have extracellular bacteria, and within intracellular bacteria, there's two different types. So we have obligate and facultative intracellular bacteria. So with extracellular bacteria, these replicate they replicate in the host, but in extracellular spaces, such as mucosal surfaces, vascular, lymphatic, and body cavity fluids, and in interstitial spaces. So some examples include Staphylococcus aureus, Pseudomonas aeruginosa, and E. coli. So these are bacteria that replicate outside of the cells of the host. Now, obligate intracellular bacteria are really parallel to viruses in that they depend on host cellular factors for replication, and they cannot replicate outside of host cells. So this is why they're very, very similar to viruses, and as you will see, viruses and obligate intracellular bacteria launch very similar immune responses towards both of them. And some examples include Chlamydia trachomatis, Mycobacterium leprae, and Coxiella burnetii. Now, facultative intracellular bacteria can invade host cells when it gives the bacteria a selective advantage. And this means that they can replicate both inside host cells and outside of host cells, depending on the host, depending on the situation. And so they kind of are able to experience the best of both worlds. And some examples of these include Legionella pneumophilia, Mycobacteria tuberculosis, and Listeria monocytogenes. Now that we have a very brief overview of bacteriology, let's dive into the immune responses against these pathogens. And let's start with the innate immune response, where we'll talk about antimicrobial peptides, pattern-associated molecular pattern, or PAMP, recognition. The role of phagocytes and ILCs will then follow. So antimicrobial peptides are really critical in the first wave of immunity against bacteria. And antimicrobial peptides are generally produced by the innate immune cells that are first on the scene during an infection. So these can include bacteria or neutrophils that have different types of antimicrobial peptides, but that act both to kill as many bacteria as possible. So for example, if we look at bacterial cathelicidin, this molecule acts by overlapping the bacterial cell membrane, seen here, in a carpet-like fashion, and then this leads to the membrane dissolving and disassociating into micelles, seen here, and this leads to the permeation and degradation of the intracellular components of the bacteria, and this kills off bacteria in that fashion. Neutrophil-produced defensins act slightly differently in that there's an electrostatic attraction between the defensin and the electric field of the lipid bilayer, and the defensin will then enter the lipid bilayer of the bacteria, leading to the formation of pores in the bacterial cell wall surface, leading to the lysis of the bacteria. So this is just two examples, and other antimicrobial peptides also function to lyse as many bacteria as possible in the initial stages of the immune response against bacteria. So how is the bacteria actually recognized? Well, bacterial PAMPs, or pathogen-associated patterns, are mostly prokaryote-specific macromolecules that eukaryotic organisms do not make. And bacterial PAMPs can also be nucleic acids, depending on the bacteria. So here's a chart with some example of bacterial PAMPs and PRRs that recognize them. So you can see that there are some nucleic acid-specific PRRs that recognize bacteria. So this is single-stranded RNA by TLR7 and unmethylated CpG dinucleotides by TLR9 within the endosome. But we also have a variety of cell surface PRRs that recognize the outer membranes of bacteria. So for example, do you remember how I said gram-negative bacteria are coated with lipopolysaccharide? Well, lipopolysaccharide is not made by host cells, and thus it's actually a very critical PAMP that activates the immune response via TLR4. This is one of the most studied TLRs. Flagellin, which are the tails on many bacteria, are recognized by TLR5 on the cell surface of cells, and different types of lipopeptides are recognized by TLR1, 2, and 6. We also have a variety of different cytoplasmic PRRs that recognize bacterial PAMPs, including NOD1 and 2, which recognize different types of acids and nucleic acids, and we also have CLE CSF8, which is a CLR, which recognizes trehalose dimycolate in the cytoplasm. So upon recognition of a bacterial infection by recognition of PAMPs, a very large innate immune response is launched against bacteria. And one of the major players of antibacterial immunity are phagocytes. Phagocytes are very critical in two major aspects, and that is in phagocytosing and breaking down bacteria, seen here. So these phagocytes are phagocytosing bacteria and breaking them down in their phagolysosomes. So they're basically eating up as many bacterial pathogens as possible. And they're also really critical in acting as antigen-presenting cells, or APCs. So APCs, as you know, such as macrophages, will present the antigens of the invading pathogen on their MHC class I molecules to CD8 cells and on their MHC class II molecules to CD4 cells. And these APCs are very critical in creating a bridge between the innate and adaptive immune responses during a bacterial infection. Now, another group of cells that are really critical in fighting bacterial infections are ILCs. Now, type 1 and type 3 innate immune responses are launched in response to bacteria. Type 1 immune responses, which include NK cells and ILC1s, are very critical in battling intracellular pathogens, including intracellular bacteria, both obligate and facultative. And ILC3s and LTI cells, which we won't talk about today, are very critical in launching immunity to extracellular bacteria. Now, type 1 innate immune responses are induced by IL-12, and type 3 are induced by IL-23. And they also produce different effector mechanisms. So seen here is our type 1 response that is mediated by NK cells and ILC1s against intracellular bacteria, and both of these cells will produce large amounts of interferon gamma, which activate macrophages to stimulate increased amount of phagocytosis of pathogens and antigen presentation. NK cells and ILC1s are both cytotoxic cells, which means they kill potentially infected cells. Now, I'm not going to be speaking today about ILC1s, but I want to go through the mechanism of NK cell infected cell recognition. So when a host cell is infected by an intracellular pathogen, such as bacteria or viruses, they change the markings of their cell surface. So to understand how that can activate an NK cell, let's go through what happens in a normal situation. So NK cells have a variety of different receptors on their surface, including inhibitory receptors and activating receptors. NK cell activation depends on this balance between the inhibitory and activating signals. The major inhibitory ligand for NK cell inhibitory receptors are MHC class I molecules. So MHC class I molecules are expressed on all nucleated immune cells and general cells, and they present intracellular pathogen epitopes. However, the presence of MHC class I molecules sends an inhibitory signal to the NK receptor, signaling that it is a cell that's either hasn't downregulated MHC or is a self molecule. And these inhibitory signals will repress any activating signal such that the NK cell doesn't kill the uninfected or normal cell. Now, during an infection, oftentimes cells will downregulate MHC class 1 molecules, or they'll present a different one, such that missing or absent MHC class 1 molecules cannot interact with the inhibitory receptor of the NK cell to lead to an inhibitory signal, such that the activating receptor signal of the NK cell is the most strong, and this leads to the apoptosis of the infected cell. Now, it's really critical that cells are killed via apoptosis as opposed to other forms of cell death, as this prevents the release of possible intracellular pathogens. NK cells can also be activated via activation of the TNF family ligand trail on their cell surface. This can bind to DR4 and 5 on some of cell targets, which will then activate caspase-6 and also lead to apoptosis of target cells. Now, type 3 innate immune responses are really critical in battling extracellular bacteria. So we just talked about innate immune responses to intracellular bacteria, so now we're shifting to those that replicate outside of the host cell. The main innate lymphoid cell that's critical in this response are called ILC3s, which are activated by a combination of IL-7, 23, and IL-1, and these produce IL-17 and 22. And IL-17 and 22 are really critical in leading to the activation and hyper-recruitment of neutrophils, which are very critical in directly killing bacteria by generating a large amount of antimicrobial peptides. In fact, you probably know about neutrophils based on pus that is created during some infections. That pus that is generated during bacterial infections is actually dead neutrophils because they're the most abundant cell during the innate immune response to bacteria, but they die very, very rapidly. So there's often a huge buildup of dead neutrophils, which is actually just the pus that you see during infection. Now, Group 1 and Group 2 ILCs work together to battle intracellular and extracellular bacteria, with Group 1 ILCs being very critical in the response to intracellular bacteria, and these lead predominantly to the production of interferon gamma, stimulating macrophages. And Group 3 ILCs are very critical in generating IL-17 and IL-22 to lead to neutrophil recruitment and active activation. So now that we've covered the major components of the innate immune response to bacteria, let's jump into the adaptive immune response against bacteria. Now, the major players depend on what kind of bacteria you have. And as we went through the different innate immune responses to intracellular versus extracellular bacteria, we're also going to go through the different responses, the different adaptive immune responses to intracellular and extracellular bacteria. So let's start with intracellular bacteria. So type 1 adaptive immune responses to intracellular bacteria depend on both CD8 cells, which recognize MHC class 1 peptide complexes, and it also depends on CD4 T cells, which recognize peptides that are presented on MHC class 2 molecules of antigen-presenting cells. Both of these are very critical in fighting responses to intracellular bacteria. So type 1 cell responses to intracellular bacteria have a variety of different roles, and I want to go through a couple of them. So on our far right of this figure, which goes through a lot of details, which would be a really good reference for you to utilize, we have Th1 cells that produce interferon gamma and CD40 ligand, which then induce and activate M1 macrophages. So this allows for macrophages to be more substantial in their ability to engulf and kill bacteria via phagocytosis. So they also induce the apoptosis. So Th1 cells are also very critical in inducing the apoptosis of macrophages that have a lot of bacteria in them. So when macrophages are going around scooping up bacteria, these Th1 cells are very critical in inducing cells that have a lot of a bacterial burden in them. And then IL-2 is produced by Th1 cells to help in activating naive CD4 and CD8 cells to allow for a much more robust adaptive immune response. And then Th1 cells also produce IL-3 and GM-CSF to stimulate production of monocytes in the bone marrow. And monocytes are often the precursors of macrophages. So the stimulation of monocyte production in the bone marrow leads to more monocytes in the bloodstream. And once monocytes enter tissue, specifically the tissue of infection, these monocytes will differentiate into macrophages, which will then aid in the immune response. Th1 cells also produce TNF alpha and LT alpha, which act on blood vessels, and they'll actually lead to the ability for macrophages and other cells to leave the blood vessels and enter the tissues more readily and more easily, because we want as many macrophages and other phagocytes to be at the tissue of infection as possible, such that there is a much higher host cell to bacterial ratio. Th1 cells also induce CCL2, which is a chemoattractant for monocytes. As I mentioned before, monocytes are critical in a bacterial infection for clearance. And so CCL2 will act as a chemoattractant for these cells once they leave the bone marrow. Now, I'm not going to go into the details of how cytotoxic T cells work because that mechanism has been gone through in previous lectures. However, cytotoxic T cells act similarly to NK cells in that they kill infected cells by recognizing the viral or intracellular bacterial epitopes that are presented on the cell's MHC class I molecules.